Hi everyone, it is my pleasure to welcome you all to this special webinar on model-informed drug repurposing applications for COVID-19. Now this happens to be the fifth in the series of webinars organized by Pharmacometrics Africa, which is a non-profit company with the goal of building pharmacometrics capacity in Africa. Today's webinar is co-hosted with Certara, the global leader in advancing modern efficient drug development. My name is Adeni Olagunju, and I'll be your moderator. I happen to be a senior lecturer and a Wellcome Trust Fellow, as well as an affiliate of the African Academy of Sciences at the Obafemi Awolowo University in Nigeria. I hold a PhD in pharmacology, and my primary research interest is infectious diseases pharmacology. It is my pleasure to introduce our panel of speakers for today's webinar. On our panel of speakers, we have Colin Pillay, we have Samuel Muksase, Yuan Cheong, and Michael Dodds. Colin is the CEO of Pharmacometrics Africa, and he also runs the CP Plus Associates. Both CP Plus Associates and Pharmacometrics Africa are social ventures that support institutions and individuals in low and middle income countries to develop scientific capability in drug discovery and development. Previously, Colin worked as a pharmacometrician and a senior leader at Roche and Novartis. At present, Colin is honorary professor at the University of Cape Town and honorary fellow of the Royal College of Physicians and a senior advisor on capacity development for global health to the Bill and Melinda Gates Foundation. Samuel is a senior director of integrated drug development at Certara. There, he is responsible for implementing model-based drug development for regulatory submissions and decisions. Samuel is currently an associate professor of pharmacometrics and clinical trial design at Université de Montréal and Lebanese American University. Yuan is director of integrated drug development at Certara. She has a very in-depth hands-on experience in PKPD modeling, clinical trial simulations, and systems pharmacology in various therapeutic areas, including infectious diseases. And finally, Michael Dodds. Mike is Executive Director of Integrated Drug Development at Certara, and he has more than 15 years of drug development experience, applying pharmacometrics with an emphasis on decision quality and business impact. Today's webinar, we start with an introduction on the current state of COVID-19 clinical research. And after that introduction, we have an overview of model-informed drug repurposing, compound screening, in silico workbench, and examples. And finally, we'll conclude with questions and answers. Before I hand over the mic to Colin, who is our first speaker today, please allow me to run through the following housekeeping items. First, everyone except the presenter will be muted, and this is in order for us to prevent interference during the session. If you have any questions during the webinar, please use the Q&A button at the bottom of your Zoom window. This will be addressed as we go along, as well as during the Q&A session at the end. You can post any additional questions you have at the end of the session to the forum section of clinicalpharmacology.com, the forum section of the clinicalpharmacology.com website. If you need technical assistance during the session, please use the chat feature at the bottom of your Zoom window to contact the panelists. Today's webinar will be available on demand after the live session, and this will be posted on Pharmacometrics Africa website, along with any resources that may be shared during the session. And lastly, we'd like to encourage you to please share today's webinar with your social networks once you have access to the recording. So without any further delay, I will now hand over the mic to Colin. Colin, please.
Thanks, Nee. Samir, I'm going to ask you to change slides for me. Colleagues, this is really, really a nice time to be a scientist. It's also a scary time. So because it's going to be a scary time, I'm going to switch my video off and go from time to time between video and the screen. So I'm thrilled to be on this panel with Nee from Nigeria, Samir from Lebanon, and with Yuan and Mike from the US. I will take the next few minutes, under 8 minutes or so, to just review a few basic clinical pharmacology principles. Most of you know this already from your undergraduate days. My colleagues from Certara will then use this as the basis to go into showing you real analyses that they have been doing in the past 8 to 10 weeks. First, a few contextual reminders. Take a look at this very familiar graph on the left-hand side of the screen right now on why we need to flatten the curve of new cases. In the first webinar from this COVID pharmacology series, you'll see it on the COVID pharmacology website. Dan Hartman from the Gates Foundation reminded us that the horizontal line that you see on the left-hand side of the screen that's labeled healthcare system capacity depends on the country or the setting in which you are. Given my personal focus on low and middle income countries, we know that this line on the healthcare system capacity is going to be rather low in the low and middle income countries. On the right-hand side of the screen, fortunately, you see that the burden of COVID disease in Africa as an example of being host to many low and middle income countries is still low, as shown by the Africa CDC dashboard. And I looked at this just a few days ago, so I guess the numbers are still valid. But all experts agree that this is temporary. So I started by saying it's a nice time, but it's also scary. So why is it nice? Well, the next slide, Samir, the next slide shows that the scientific community is delivering. The picture on the left-hand side is the clinical trials on the Cytel clinical trial tracker. Now, I did this screenshot on the 8th, it's shown there. If you go now, you'd probably see that the number has gone up already. I'm convinced of that. So this is fantastic. And look, even Africa is represented. And we all know that Africa is traditionally home to only 2% of global clinical trials, despite being home to 1/7 of the world's population. So the scientific community is delivering. And then on the right-hand side, this is from The Economist on Friday, there's an exponential increase in COVID-19 scientific papers. Wow. Up to just under 7,500 accessible papers in a system that if you have been trying to publish papers, you know that this usually takes months to years for the scientific community to release its findings. So we're getting information out pretty fast. So why is this scary? With the proliferation of trials, we have already seen examples of poor scientific rationale for the studies. With the rapid release of scientific information and it becoming available so quickly and accessible to the general public and to our politicians, it means that expectations are being created that we as scientists are going to have difficulty being able to fulfill. Okay, so with the proliferation of trials and information becoming available quickly, we are having expectations that we need to fulfill. So now more than ever before, we as scientists need to stay on top of the emerging knowledge and to put the emerging information into context, reminding people of the iterative scientific process of an idea that somebody has, converting that into theory, stating a hypothesis, generating the evidence, and then putting in recommendations and policy. In the advertisement to this webinar, we use the word chaos, and maybe that's a bit harsh. So can we transition from chaos to coordination? So there's a lot going on, and there is a need for coordination. Multiple groups have stepped up to play a role, and I think this is a great thing. On the slide right now are at least 3 initiatives that I'm aware of and I'm involved with. I would also ask you at this point, I'd invite you to use the chat feature on the Zoom and type in any other collaborations that you're aware of, any other collaborative groups that want to come and get people together in order to get to some stage of coordination. So we do need to transition from chaos to coordination, and we need to make people aware of what's going on. Next slide, please. So here's my few slides on clinical pharmacology. I'm talking to you right now from Basel in Switzerland, and here Paracelsus, a Swiss physician, spent a lot of his scientific life. And we often quote his famous words, "All things are poison. Only the dose permits something not to be poisonous." I like to think of this as the tenet of clinical pharmacology. It's all about dose and response. In the next two slides, I'll mention pharmacokinetics and pharmacodynamics, and I see these as bridging disciplines. The people like myself who use mathematics in order to quantify the processes create the science of pharmacometrics. My main point here, however, is that to be effective, we need well-designed studies to address the research question at hand, good data, well-executed clinical data collection, good laboratory analyses, data managers, and a slew of other disciplines in the clinical science spectrum. In brief, clinical pharmacology is a team sport. Very rarely will you have a clinical pharmacologist that has the luxury of all those skill sets in one person or within his or her clinical trial unit or university unit. Next slide, please. In pharmacokinetic studies, we measure drug concentrations at various time points to characterize absorption, distribution, metabolism, and excretion, the processes that make up pharmacokinetics. Our eventual goal is to use concentration as a surrogate to relate to drug effect. If the study has rich enough sampling, we could directly read out, as you see on this curve here, the maximum concentration, the minimum concentration, or the area under the curve, sometimes which is a reflection of the average concentration, or any metric that you want to use on the concentration versus time curve to relate to drug effect. Now here, let me cite two examples of why I say this is a team sport. In pharmacokinetic studies, actual time of dose and sample collection needs to be meticulous. Those of us that have analyzed data collected at a different unit know the white lie that my study team always collects samples as per protocol. However, if we are collecting samples in the rapidly changing absorption stage, and Samir, you could point that out on this curve, or at the end of the dosing interval and didn't have an accurate dosing history, then the data is essentially uninterpretable. Another example, some drugs like chloroquine will concentrate inside red blood cells. And we know that this is up to 3 times more than in plasma or serum. So the lab analysis needs to develop a precise and sensitive assay in the sample matrix that makes the most sense, or advise on appropriate correction factors based on partitioning. We usually measure in plasma or serum, not at the site of action. Since COVID-19 is a respiratory problem, we don't measure in lung, we need to figure out how to relate the pharmacokinetics that we measure in blood to the concentrations in lung. So in addition, on the previous slide, when I mentioned that we could just simply read off pharmacokinetic metrics, one could fit a mathematical model to the data. This would allow one to reduce all of those concentration time curve points, and there's quite a few on this particular curve, and we don't often have the luxury of that, into something that could be used, parameters like clearance, volume, and absorption rate, as shown on this PK model. And using that model, then predict new scenarios like full profiles from sparse data, alternate dosing regimens, alternate untested situations, all of that contingent on the data that was used to build the model. Physiologically based pharmacokinetic models, another type of model, Johan will mention this briefly, can relate drug concentrations measured in one tissue like blood to other tissues like lung to give more insights into effect site concentrations. In the rest of this webinar, you will see simulations of drug concentrations from dosing regimens from PK models. Let's go to the next slide. So from pharmacokinetics to pharmacodynamics, the picture that you see on the left-hand side is a model that we often or very frequently use in pharmacology, the Emax model. If you follow a line on the y-axis is effect, and the effect is either a beneficial effect, like resolution of the symptoms of the disease, or a side effect. So if you follow the line on the dose or the concentration axis, the x-axis, you will see there's, as dose or concentration increases, nothing happens until suddenly you start to reach a threshold and the effect increases up to a maximum or 100% of the effect. A key point here is the concentration at half maximum effect that we call the EC50. Now, in clinical pharmacology, all clinical pharmacology processes follow this process of having two curves, one on the left shown on the right-hand side graph, the blue curve, and that's your beneficial effect curve, and one on the right, which is the adverse effect or tolerability effect curve. For the clinical condition under consideration, you will need to determine or define what the effect is. So for example, in malaria, it might be resolution of fever. And if you're using a drug like chloroquine in malaria or a drug like chloroquine in a rheumatoid disease, the effect would be defined differently. For COVID-19, we have not yet defined what the shape of that curve looks like yet. The adverse effect curve, however, especially for repurposed drugs like the ones that will be covered in this webinar, is usually transferable from one clinical condition to the other. Oftentimes what happens is that, well, in early drug development, we usually have the luxury of defining what this adverse effect curve would look like in a somewhat systematic way with the single ascending dose and multiple ascending dose studies. Thereafter, we depend on reported adverse effects. So for COVID, we are likely to test higher doses, and again, getting to within the beneficial effect and adverse effect curves is going to be the challenge of what we need to define in clinical pharmacology for therapeutics for COVID-19. One more point before I leave this slide. You will see similar shaped curves like this, the sigmoidal curve shaped curves, derived from laboratory experiments where we are looking at the killing of the virus, the killing curves, and you'd see similar parameters of EC50 and IC50. Our goal with COVID therapeutics would be to find a concentration in plasma or blood that is several times higher than the measured concentration in the lab for the virus. Staying within the range of efficacy and safety is the therapeutic window. Next slide, please. Back to my point about this is a team sport. Success with any of the therapies is going to need multiple contributors. When we give a dose and we expect an effect, as shown on the two red boxes on the left and the right of the screen, in between there's a whole slew of processes that are happening. A plasma concentration or blood concentration measurement, an effect site concentration, and then a whole series of effects on the target before the response happens. For success to happen, you need all of those processes to work. For failure, arguably, it might just be one. Last slide, please. Back to my excitement about where we are. We are going to conquer this problem. COVID-19 has several potential druggable targets. These are two very recent papers. The one on the left very nicely depicts the targets and the drugs under investigation, some of which will be described in today's webinar. The graphic on the right shows, from that reference, shows the drug therapies that are currently in clinical trials. And the numbers that were there on the connection lines shows the number of therapeutics investigating those drugs. With that, let me hand over to the next speaker, Samir. Thanks very much.
Thanks, everyone. This is Mike Dodds. I'm going to spend maybe 10 minutes introducing you to what we think of as model-informed drug repurposing. In the context of a novel pathogen like SARS-CoV-2, the point of repurposing existing drugs is to offer a promise of some kind of rapid treatment response to the disease while we develop more specific treatments. We don't think of repurposing existing pharmacologics as a panacea, right? These drugs were never designed to deal with this particular virus or coronavirus of any kind. But this does offer us something we can do right now while we develop more specifics and presumably more potent molecules. Model-informed drug repurposing, MIDR, approaches are thought of in the same sense as model-informed drug development. And very broadly speaking, model-informed drug repurposing is the application of a wide range of quantitative tools in drug repurposing to facilitate prioritization and decision-making. So you can think of this process as a filter, although that isn't entirely accurate. It's more like a method by which we can prioritize these precious resources that we have. And in terms of resources here, we're not talking about sponsors' dollars or that sort of thing. We're talking about time and patients at risk, right? Every day that we go through this pandemic, there are patients in need of some kind of potentially life-saving interventions. And in terms of opportunity cost, if you offer them the access to a repurposed drug and it turns out to not be terribly effective or perhaps not beneficial, actually, more risk, then you've denied them the opportunity to potentially access another repurposed drug which could be beneficial to them. If I were to try to give you, for those of you who spent time in traditional drug development, to give you kind of an analog or how it feels to be working in this space, which is very rapid and very pertinent, it feels something like an oncology indication where we have a poor standard of care, right? In that situation, we're always thinking about risk-benefit of those patients. Perhaps earlier in their cancer, we may be more risk-averse, right? They have many years ahead of them. Later in their disease progression, we might be relatively risk-seeking because there isn't a ton of other options or time left to them. And that may be mirroring how we treat people who are early in presentation of symptoms versus patients that are heading towards the ICU and intubation. The other area where this feels relevant around oncology is the concept of adaptation, right? Oncology is an indication area where they've really done a great job of learning as they collect data, confirming that with steadily larger trials, and then adapting, pruning out the dead wood, if you will, and focusing on getting to the good stuff, if you will. So how is this working? Well, we're trying to develop a therapeutics clearinghouse, and really what I mean by that is a scientific fact base, right? It's trying to collect information on pharmacokinetics, pharmacodynamics, what information we can find out about the viral life cycle, the epidemiologic modeling that's been going on, viral kinetic models, right? Bringing all of that information together into an in silico workbench space where we can combine these ideas and try to use them in a triage type of environment. I spoke to the concept earlier of cost benefit. We have lots and lots of drugs that we could potentially repurpose, right? Potentially any drug that we've made in the past, and it could be useful against this virus. So there's a universe of potential options available to us. And so we're paralyzed with the number of options that we could go forward with. And the idea is to use these tools to start with a very wide funnel at the beginning, and use that information to kind of separate the wheat from the chaff, if you will, right? And my two colleagues, Samer is going to talk about that early filtering or triaging strategy. You know, how do we deal with the universe of potential options and try to filter it down to a dull roar, if you will. And then Yuan is going to continue on from there once we've identified some molecules that we think are potentially beneficial, working those up in greater detail. These ideas are not new. I can give you an example from traditional model-informed drug development, where we've developed vaccines and treatments or small molecule interventions for seasonal influenza, right? And then try to apply that to a novel influenza strain where we're doing model-informed drug repurposing. So the bottom of this diagram should be pretty familiar to you, right? This is your classic drug development process, right? You have an indication in mind, seasonal influenza. You begin working up your in vitro and in vivo virology information, work through preclinical analysis, and then go through your classic clinical phase 1, phase 2, phase 3, and then move towards approval. And as you're doing that bubble here is the model-informed drug development portion of it, where we're building relationships through phases of development, and the information base that we're gathering begins to gather steam, begins to synergize, and gives us more information as we carry through early initial in vitro data all the way through to registration. So that's been very beneficial to normal traditional drug development. In model-informed drug repurposing, which is the top of this slide, in that case, we might have a novel influenza strain, and we're going to generate probably in vitro and some in vivo virology information. But then we're going to very rapidly progress, probably potentially skipping a lot of preclinical activities. We're going to go immediately to anecdotal clinical data, which, Colin showed you all of those trials that are kicking off. You can't imagine all of those trials are, randomized placebo-controlled trials, right? They're going to be very small trials, potentially done at one clinical site with perhaps not the best set of controls, but it's the data you have, right? And the critical link here is this bubble in the middle, where we take that in vitro and in vivo virology information, we gather up the anecdotal clinical data, and we merge that to leverage what we understand about the drug that's been developed for classical seasonal influenza in this case, right? So these are not new ideas. We're trying to leverage what's already in our hands, right? So the approach that we're taking from model-informed drug repurposing proceeds in stages, and it really builds as the information about this novel pathogen, about the disease that it causes, begins to build. Today, we're going to talk about the first two stages of this process, which are the first two line items in this diagram. The first is, at the very beginning, what do we have? We have probably viral information around the in vitro pharmacology of this particular virus. And we have labels for all of the drugs that exist in our hands. And we can start asking some questions around, some very simple strategies around, well, do we think any of these drugs can achieve concentrations that matter, at least in the test tube, right? And Samara is going to walk you through that initial screening situation at the top of the slide here. Then as we detect molecules that potentially are particularly good, or in one case we have today, particularly bad, we think, we then begin to work that up a little bit more detail, right? We take that in vitro pharmacology and we marry it to the pharmacokinetics that we understand from the label where the drug has been used in clinical practice. And there we're thinking about adjusting posology, right? There's nothing saying that the drug dose that was used in, say, HIV is in any way relevant to the treatment that we should take for this novel coronavirus. What's not covered in the presentation today, and maybe we can commit to coming back in 6 months' time, again, when we have more information about this particular disease, is then beginning to build out as we start getting clinical pharmacokinetic information, right? We start collecting blood samples in patients that are treated with these repurposed drugs. And we start bolting that to the virology information, right? We're just at the cusp of being able to do that now. We've got maybe 50 patients worth of viral kinetic data, that sort of thing, right? And there we start moving into, you know, what are the exposure predictors of viral dynamics, right? And then that builds into looking at clinical pharmacokinetics and then clinical outcome, right? There's going to be a wave in the next few months of all of these, you know, hundreds and hundreds of trials beginning to read out. And there's where model-informed approaches like exposure response analysis can be particularly useful to detecting signals from these positive or negative from these clinical trials. And we can start asking some insightful questions like, well, is this disease a special population? Is there something about the disease that's fundamentally changing how the drug behaves in the body, right? That could certainly happen. Then finally, as a capstone to these activities, we're going to start reading out these very heterogeneous study designs. Traditional clinical trial analysis techniques really struggle with trial heterogeneity, right? What else is going on in the background? There's evolving standard of care, right? We're getting better at being able to stabilize and treat these patients, right? So the mortality rate is shifting underneath us as we move through time. The clinical population may be shifting as well, right? And then additionally, the pathogen itself may be mutating, and so the body's response to it can be changing over time. Again, traditional analytical techniques that look at t-tests, that just look at one particular study, are really going to have a hard time and struggle with this level of heterogeneity and shifting sands, if you will. And model-based meta-analysis is particularly suited to be able to handle that. So that's my introductory remarks to give you a sense of what model-informed drug repurposing is. I'm going to turn that over to Samara now to walk you through the first portion of this, which is analysis of the existing label information.
Hello, thanks, Mike. Hi, everyone. Hello to all friends and colleagues all across the world. I was very pleased to see familiar names and new names on the participant list. So as Mike introduced the topic, the first part that I will be presenting is about the first layer for triaging the compounds of interest. Basically, we look at in vitro information, we collect the label for the repurposed, hopefully repurposed drug, and we look at the metric we are calling proof of hope of this compound working against COVID-19. So basically, we have two different metrics shown on this slide, on the left and on the right. And on the left, it's about the knowledge we have from an already approved on the market drug about its indication, approved indication IC50. And then we compare it to the COV IC50 in vitro. That's an easy metric to collect. Just to illustrate, I'll be back to the slide. But here, hopefully we have a PK profile. We know about the label IC50. So usually when we approve a drug, the concentration we achieve are maybe in the middle, maybe a little bit higher than the label IC50. But for sure we are in the range, because to be approved, we will have some kind of efficacy. So that's the first metric. And then second metric, as we know more, like it's more elaborate here, this is C-state average, because...
Samir,
yes?
Samir, sorry to interrupt. This is Colin. We're getting some messages coming through that the audio quality from your side is a bit low. I would suggest maybe that you also get rid of video and see if there's anything that you could do to improve that. And Gabe, if you could try and help as well.
Okay, yeah, video is off, and I hope it will be better. So just as a reminder quickly, so I was talking about label IC50, and see steady state average. And I'm trying to get it. Okay, so basically we have an approved drug, and we usually know the PK information about already on the market drug, and label IC50, like for the approved indication, we have some kind of a PKPD knowledge, and we know at what concentrations we have an effect for the previous indication. And as you might have seen, we have a lot of information pouring from everywhere in the world about the activity of XYZ drug in vitro on SARS-2, on the virus causing COVID-19. So, and you might be surprised that many times, especially for oral drugs like chloroquine or hydroxychloroquine, we don't have a good knowledge of like an approved label IC50. And for some of these drugs, we might not even know how they work in detail. And the second step would be to look at the profile and look at some kind of pharmacokinetic summary, whether it's a Cmax or Cmin, as Colin mentioned, or a C average steady state. So here we are showing a single dose profile, but with multiple dose, we have some accumulation, and we look at what we will achieve at steady state. So of course, our premise here is we quickly compare the label IC50 to the curve IC50, and if the label IC50 is like similar or not, basically we compute a ratio if it is within 5 times. So if the curve IC50 value is lower, that means we will achieve concentration much higher than the IC50 and will achieve efficacy. We will kill the virus, at least in vitro. And then if the curve IC50 is much higher than the label IC50, we are unlikely to kill the virus in vitro, let alone in vivo. But that's like a quick way to triage. And of course, you will see later on with my colleague Yuan, will have to take into account more information about ADME, about unbound fraction. We will use even a full PBPK model in some instances. But this first step, we want to prioritize and quickly know which drugs will likely have an effect on killing the virus. So this is effort is not to reject a compound, it's really to prioritize which one we will build a full model or a full simulation dashboard to see what are our chances of responding. And so basically to do so, on our covidpharmacology.com website, we have a toolbox of tools that everyone can access from the website. And I will be walking through the compound screening dashboard. So basically in this dashboard that you can access, we have some background info, some science behind what we are doing, such as the slides that Mike described earlier. So we'll include all the essential info for the researchers to triage the compounds. So here I have some screenshots, and when you see a red dot, that's like you clicked on this part of the dashboard. So here I clicked on clinical pharmacology triage. I want to select like compounds of interest. The usual suspects we all heard about in the news, maybe chloroquine, hydroxychloroquine, other like ivermectin, is it working, is it not, etc. So you select what you want, and you can further drill down and filter by mechanism of action. And then you will be presented with a table that has a lot of info. It will have molecular weight, it will have EC50, it will have ClinPharm detail like approved dose, like dosing interval, and so on. So you can select a bunch of compound of interest, or you can select all. So here I selected all that I selected as a previous step. And once you select the compound, a heat map will appear. So that heat map will have each compound, chloroquine, glucosamine, etc., presented, but also we will have a ratio of the Cmax according to the approved dose and known PK. So we know what would be a Cmax. We compute a ratio with respect to the EC50 of its activity in vitro. And then Cmax, Cmin, and the primary indication is 50. And as you will see, many of these compounds we don't have easily the primary, quote unquote, primary indication is 50. So you will have a lot of gray boxes. And that's why we are using multiple metric. And just as an example of a consensus metric, I computed a mean of these different ratios. So what's important here with this heat map is you can easily choose by which metric you want to order and sort the heat map. So the default is the mean ratio metric. When you click of, say, I want to sort by Cmin EC50. So you see here, the order changed, and I selected good compound on top. So these have a better ratio. And you see that chloroquine, hydroxyl, okay, so order changed because I select Cmin. When I compare here, it's ordered by the mean ratio. So it will help you quickly sort and reorder the way you like. And if you want to change the order bad on top, so keep the good down, that's an option. So I try to include as many options as to keep the user comfortable in the way and the color he likes and the way of ordering we want. So as a step back here, I selected all the 23 compounds we have in the small the database that we keep building and evolving. So, and I'm sorting by mean ratio metrics. And here I can zoom. That's an interactive for interactive plot. You can just select the region of interest and you zoom into it and you highlight, you have some hover effect. So each cell here you will have like the ratio value, not only a color map. Color is good to characterize, but you can also look at the number in detail. So, of course, we will also link to the full simulation dashboard once we have them ready. So there will be a way to go directly to the other tools that my colleagues will be showing later in this webinar. And as I mentioned earlier, this table here, you have a lot of info about the ClinPharm, but also you will have a section here called full clinical pharmacology tables that my colleague are working on. And then you will have really detailed view of everything important we know about the given compound, whether it's like PK, PD, DDI, currently running trials. They will all be available under this tab. So that's all on my end, and I will hand over to my colleague now to continue.
Thank you, Samuel. Hello, everyone. This is Yuan. Hope you can hear me clearly. Okay.
Yes.
So great. Thanks. Okay. Like Colin mentioned at the beginning, this is a special time for all of us, and although we all stay at home, hopefully most of us are staying at home. either giving this webinar presentation or listening to it, I think probably most of the people in the audience and we are focused almost on the same thing, the common enemy to the humankind at this moment. For this section, I'm going to focus more on our Silico Workbench that integrates in vitro potency data with pharmacokinetics profiles of the repurposed drugs. Okay, so first, I'd like to provide an overview of our approach. So we consider this approach as briefly introduced earlier already by Colin and Mike, and also briefly by Samir, where we combine in vitro data with PK knowledge is most useful for early posology assessment and also able to provide insights in certain aspects of the study design. So here we mainly use the established PK models for the repurposed drugs. Usually they are population PK models, also we call PK, and physiologically based PK models, which is PBPK. We use these two types of models most often. Luckily for the repurposed drugs, they are generally available and readily accessible in the public domain. So these two types of PK models, they have their own pros and cons as listed here. So for PK, usually the level of between subjects variability are well understood. Also, the major influential covariates to the exposure levels were usually thoroughly characterized and studied already. Also, the mathematical forms for these PK models usually are straightforward enough for us to implement in simulations. However, PK models usually focus on only plasma or serum concentrations and lack concentration predictions at specific sites of action. In the COVID example, we are in particularly interested in the concentration at lung tissue. So these are some of the limitations from the PK models. On the other hand, we have physiologically based PK models. These models, they integrate more mechanistic understanding of human organs and the physical chemical knowledge of a compound. So usually they provide better handling of tissues or sites of action with a higher granularity. Also, due to the nature of mechanistical understanding, the PBPK model also can handle special populations from more mechanistic approach in comparison to the empirical approach used most often by PK in these situations. However, for the PBPK models, their results are often in a more complex form and more difficult for us to export to external simulation tools in order to run real-time adaptive live simulations. So these are the pros and cons of these two types of PK models. In the current situation, though, each of these two types can be used depending on our application-specific repurposed drug. We can use them to get a reasonable approximation to our critical question, which is what is the dose exposure relationship of this repurposed drug at a relevant site of action, and how does that compare to the in vitro values that are thought to inhibit SARS-CoV-2. I saw some audience has asked questions regarding in vivo, IC50 in vivo potency values. Unfortunately, at this moment, our most knowledge are still focused on in vitro values. We basically acquired from the laboratory in different cell lines. So, but we believe with the more data coming out, we might get better understanding of the in vivo potency values for those drugs. So in order to really answer this question, when we use the PK model, usually we will add, for example, unbound fraction calculation for the drug in the blood. And we also, sometimes we will use published tissue versus blood ratio observations. Sometimes from PBPK models, we can even derive the tissue versus blood ratio theoretic values. So in this way, we kind of work around the limitation from the PubPK side. On the other hand, when we are using the PBPK model, usually we will bracket covariates with simulations and then do interpolation. And also we can use superposition of single dose simulation to simulate different posology. But apparently there are some limitations also using this approach, for example, whether there are enzyme reaction or other nonlinearity in the PK. So anyway, these are the main approaches we use to basically derive the time course or PK profile of a repurposed drug. Okay, so let me see. Okay, next slide. So before going to the specific examples, I'm going to also bring us to take a glance at how to access our workbench. By now, you probably are familiar with or have heard of the COVID-19 Pharmacology Resource Center. So yeah, I see that a few of our panelists were posting this in the chat or in the Q&A. So here I also provided the link here at this site. And if you open this COVID-19 Pharmacology Resource Center, you can see there is an in Silico Workbench tab at the top, and you can just click on it. And it will show our current inventory of in Silico Workbench. So the first one actually is what the nice tools that Mia has presented, the compound screening dashboard. And then there are 6 repurposed drugs. We already developed the tool, azithromycin, chloroquine, hydroxychloroquine, ivermectin, lopinavir, ritonavir, and tocilizumab. So these are probably familiar names to you because a lot of them are under hot debate whether they should be used or they should not be used, whether they're too toxic or whether they have any promising efficacy at all. So also we are diligently working on additionally the new ones. We are expanding our inventory for the newly emerged repurposed drugs as of now. Okay, so this is an example for the chloroquine workbench. So if you click chloroquine in the previous page, you will see something like this. At the top, you can see that we have 4 different basically dashboards, current protocols, new protocols, about references. So for current protocols, we provided options for the user to select an established regimen from labels or literature. In this case, the selected one is acute malaria, FDA, and then you're going to also see acute malaria, WHO, or malaria prevention, FDA, something like that. These were already established in the literature or publications, and at least there are some evidence to prove they are efficacious in that specific indication in the past. So also we have enabled the selection of site of action. So at the moment, the none is selected, but you can also select, for example, plasma or serum, depending on the drug. So also some other control, number of weeks of follow-up. Basically, this will show you how the exposure will change if you stop the treatment, and how long it will drop to some threshold. And also some other PD-related control. We're going to get to that in a couple of slides. So here you'll see the PK simulations, which is what we are showing, and you're going to see in our examples how this can help us to make some, gain some insights and make some decisions. Also some PD simulations and dosing simulations. In the PK simulation results screen, you can see we have integrated different targets. All this for now, as I mentioned, are in vitro targets that we have connected with the help of a lot of clinical pharmacology colleagues that from different sources of literature. Some are earlier ones, as early for this, as early as 2006 and as late as 2020, which is maybe just a few days ago. So we have put all this threshold on top of the PK profile, so you can clearly see how much the simulated exposure compared to each of the threshold. Come back to the PK profile. Here we have a central tendency, which is the predict median, and also the gray area, which is the predict 95% confidence interval. So in the next slide, I think it's a little bit too fast. Okay, so this is another screen. Actually, you probably feel this is a little bit familiar with the previous one, but notice now I'm looking at a new protocol where I can basically play with all these options and customize what dose and regimen I want to actually use to simulate the PK profile. In this case, we also allow users to select extra initial doses. Often this is called loading doses. So you can see how how this can change the PK profiles in this window in real time. So basically allow users to play some what-if scenarios investigations. Okay, in the next slide. So it's a little bit hard to acronym the control to move to the next slide. Sorry about that. But this is the next slide where we show that the PD simulation part. So there's really not too much at this moment to play with the PD, as we mentioned. The most knowledge we got are from the in vitro potency values. So here we allow users to select different PD targets, which corresponds to the literature documented in vitro IC50 or EC50 values, and we allow the user to choose how they want to visualize the buffer, which is how does the target concentration compare to the threshold ratio. So basically here we are choosing two thresholds, 1 and 5. Sorry, it's a little bit too small to see, but you can see that this shows in which time points you can have a threshold, you can have a clinical concentration or simulated PK profile that is greater than 5 times of the target. And in which of the times you're going to see that actually the percentage of concentration you're going to see, which is between 1 times or 5 times of the target, and in which of those red area you're going to see actually we're not able to achieve the target. So another way to help us to visualize the comparison between PK and the in vitro targets and have an idea of how promising this in vitro, this repurposed drug to achieve the in vitro potency. Okay, so now it's time to go over a couple of examples, and then at the end, Mike will also talk about another example before he shows some real-time demo. The first example I'm going to talk about is chloroquine. Apparently, this is something under hot debate together with hydroxychloroquine, which is thought to be an improved version of chloroquine. So chloroquine and hydroxychloroquine, actually, they have both been extensively used in treating malaria and some autoimmune disorders since 1940s. And they are both considered to have different, like multifaceted antiviral effects. Some thought they can prevent virus entry to the cell. Some also suggest that they have some effect to prevent virus replication. Anyway, so here I'm showing a few established or current protocol that's already been established long ago and implemented in our simulator workbench. So this includes acute malaria treatment and malaria suppression treatment. So another drug often used together with chloroquine and or hydroxychloroquine is azithromycin. So this one basically is an antibiotic. It's believed to suppress airway inflammations, and also it has been used for a long time. It's like 40 years already. So however, when we already know long ago, if we even just use them separately, there are some adverse cardiac events. So in particular, in patients with a pre-existing heart condition. Now there is rather famous study called COVID-19 study. It's already registered in clinicaltrials.gov. So in this study, there are two dose groups. The high dose group use a chloroquine at 6 milligram BID for 10 days, and the no dose group use 450 for 5 days. In addition, all the patients received another drug and azithromycin. So, okay, so now we have the protocols and we have the drugs. Actually, we have both chloroquine and azithromycin, but for this example, I'll focus mainly on the chloroquine. So how do the current protocol, under the current protocol, how do the exposure compare to previously established regimen? So here I'm showing the comparison. So this is on the left-hand side, you'll see the WHO acute malarial regimen simulation. So we can see that it was used only for 3 doses, and then the Cmax is around 10,000 nanogram per ml. But under our current high dose simulations, we can see that the exposure goes really high and the peak around 30,000. I know you can see if you just look at the plots, they are almost the same height, but I want to point to this different y-axis. This is 3 times higher than this y-axis. So we can see that this is much, much higher relative to the previous established acute malaria exposure. So of course, there will be some toxicity concerns. The preliminary findings from this study has found that out of a predefined 440 patient sample size, and then 80 patients were enrolled, the high dose arm presented more QTC events, and then also basically it's not safe, but there is a very high lethality. So of course, the fatality rate was high, and basically that means even if it had some efficacy, it's not basically balanced by the high toxicity it brings to the patient. So this finding suggests that the higher chloroquine dosage should not be recommended for COVID-19 treatments because of its safety hazards. So that's what everybody reads from the news. This trial of high-dose chloroquine halted early due to toxicity deaths. Okay, let's move to the next story. This is about very famous combination literature, which is the lopinavir ritonavir combination. Literature is among the earliest used repurposed drug for COVID-19. It has been approved and used for the treatment and prevention of HIV. The antiviral effect were thought were through mainly lopinavir, which the PK profile was boosted by ritonavir. So lopinavir is one of the protease inhibitors that are believed to prevent virus replication. So for the HIV treatment, literature are often used over a relatively long period of time. When you read the FDA label, you can see that it was actually approved based on studies of 72-week treatment duration. However, now for the acute virus infections such as COVID-19, the use of the antivirus could be a little bit different. So there is a relatively well-known knowledge that for antivirus to really be effective in the acute virus infection, we need to use this treatment, to initiate this treatment early on after the infection. So that tells us if we want to use this antiviral combination, it's very important to push the exposure as high and as early as possible. So now we can then explore how we want the dose to look like. So here we simulated two scenarios. Both have the same maintenance dose regimen, 400 milligram BID. The left panel shows the PK profile without loading doses. And at the right panel shows the PK profile with Q6-hour loading at the beginning. So here I just picked the highest threshold, the yellow curve, and to basically to look at when the concentration can arrive at this threshold and how long it can stay. So from the regimen without loading, we see that the concentration reaches at the target on day 2 and stays above for less than 2 days, versus for the regimen with loading, the concentration already can reach this threshold on day 1, and it will stay above for more than 3 days. So clearly, this type of literature regimen with loading doses probably will be a better bet when we treat acute virus infection like COVID-19. Okay, so that concludes the section from me, and I will hand over to Mike to talk us through another example.
Thanks, Yuan. So this case, ivermectin, is one that I'm particularly passionate about. Ivermectin is a very important compound used in the African continent. Next slide, please, Samir. It's used extensively for 5 neglected tropical diseases at single oral doses of 150 or 200 micrograms per kilogram. Those of you with calculators at home will note that that's a 14 milligram dose in a typical 70 kilogram subject. So a highly potent drug to be working at a 15 milligram dose exactly once. Recently, Calais reported that this approved drug ivermectin inhibits the replication of SARS-CoV-2 in vitro, and they found that it was able to inhibit the replication of the virus in Vero cells with an IC50 of approximately 2 micromolar. This unfortunately generated a stampede to buy up ivermectin, which is also used in a veterinary setting. And in fact, anecdotally, I can report that my sister-in-law in Denver, Colorado, who's a veterinarian, was unable to require ivermectin for her veterinary practice, right? So the problem with this issue, and I'm going to try to keep my temper under control here, was the publication then assumed that since the drug is FDA approved, this is going to readily translate into a treatment or a cure for COVID-19. The problem is what they didn't address in the publication was whether a single dose of 150 or 200 micrograms per kilogram could actually result in adequate amounts in the human body that would be able to inhibit the replication of SARS-CoV-2 in the blood or even in the lungs, right? So they simply put that marker out there to say, look, this kills SARS-CoV-2 in the test tube. But what they didn't answer was, well, yeah, but can we get the drug concentration up to those levels and do so safely? Our preliminary findings and findings of others then have suggested the standard doses of ivermectin wouldn't result in efficacious concentrations. So that you'd have to go to extraordinary doses to achieve efficacious concentrations. And if you did that, that would probably lead to unacceptable toxicity in these patients. So this is an example where, Yuan showed you in a Silico Workbench tool where we allowed and encouraged people to look at the existing regimens that people have trialed already, at least as we're tracking these clinical trials, and then allowed them to work through adjusting posology to make a trial for their particular patient population. For the ivermectin case, we don't do that. In this particular case, we don't feel that the evidence is sufficiently compelling to warrant use of ivermectin in a clinical study for any reason. When we look at, these are projected concentrations of the drug in the lung. We can also look at concentrations in the plasma. You'll notice that the y-axis is, we're not cracking 1 micromolar. In fact, we're several hundred fold below the IC50 value reported in Calais in 2020. That's the horizontal dashed lines. So, even if you were to use the drug, in this case, there's three daily doses on the far right here. This is a mosquitocidal regimen. So we're pushing the dose to have it be relevant for mosquitoes, and we just can't crack that concentration. It's just not, we're just not able to do that. So we had some questions in the Q&A section around, are these tools providing directionality? Are they helping inform what's happening in the clinical trials? And I would argue with this particular case, yes, right? This strongly and stridently argues, please do not use this important drug that's needed for people with onchocerciasis. It's a critical drug for that. And diverting that drug supply to attempting to use this in COVID-19 is both, I think, risky for those subjects who could be trying another repurposed compound, something like lopinavir, ritonavir, chloroquine, hydroxychloroquine. So it's risk to those subjects, right? The cost benefit there, they're not able to access another treatment. And then you're definitely taking away a drug which we know to work against a critical disease like river blindness. So I'm going to go ahead into the risky portion of the presentation and open up the presentation live. So this is always tricky. So I'll do my very best. So this is those of you who've been on the chat have been asking, where can I get these tools? How can I access them? They're here at covidpharmacology.com in the Silico Workbench. There's a forums section over here where we've been copying out these questions that people have had, and we're going to put those out there on that forum so we can continue the discussion, and importantly, keep track of the conversations that we're having. Colin showed you before, some information on that clinical trial tracker, and we can actually take a look at that here live in real time. And in fact, he's right. The trial count has definitely ticked up since he took that screenshot. In this in silico workbench, I think a very relevant compound to the African continent is chloroquine. And there's a bit of a bio here supplied for what's going on with a particular tool. And that allows you to then click, there's some introductory information, right? We've been able to show that it does indeed inhibit SARS-CoV, MERS-CoV, and SARS-CoV-2. So it seems to be broadly effective against, or at least in the test tube, against a variety of coronaviruses. There's definitely some protein bindings, and we have some caveats there to kind of help you understand, what we can and can't do with these simulation tools. If I scroll down carefully, this tool allows you to look at, different regimens that people have already considered as part of clinical trials. There were some questions around, how is this, these drugs are already being explored in clinical trials. How are these tools particularly useful? Well, one of the things I hope you can appreciate is when you look at how these drugs are treated, they're used quite differently. The posologies are not set in stone. We do not have a defined dose and regimen that we know to be effective against COVID-19, right? We can reuse doses and regimens they've been used for other indications, but that doesn't mean they're necessarily the right choices for COVID-19. So what we're showing here is, these are the existing regimens that have been out there. That may be okay, and it's a great starting point for people to kind of look at. I think where the interesting part is, where can, how long do I want to run my trial for? This allows people to go in there immediately and sort of say, well, what if I extended the days of treatment out to 4? What happens if I extend to, roughly speaking, a week worth of treatment? What is that going to do in terms of drug concentration? And again, I'll remind you, Yuan walked you through these different IC50 values. And what you're appreciating here is that, yeah, we're able to clear those IC50 values in this particular setting, taking care that, we're looking here in the lung, right? We're looking in the relevant site of action. That's a critical piece of information. The drug concentration in plasma is quite different, and you'll notice that, at least if you look in plasma, you would see that the drug concentration fails to achieve those IC50s. Well, this is a property of chloroquine and hydroxychloroquine, that they have a particular affinity for the lung tissue. So, we think that that's a key consideration for this particular treatment. So that's the kind of the live demo I wanted to share. We're coming up at about 10 minutes remaining. Let me turn it back over to Nii, and we can walk through some of the questions and answers. I noticed that the Q&A panel is quite large. We have probably 20, maybe 30 questions. I don't think we can get to all of them in time, but again, we're going to take those comments and move them over to the forum, and we can continue this conversation. So Nii, let me turn this back over to you.
Thank you so much, Mike, and thanks to Colin, Samuel, and Johan. We truly appreciate sharing your expertise with us. Now, quite a number of the questions that were raised during the session were answered. 15 questions were answered, but I'll just raise some of them in case you want to make additional comments, or maybe other panelists would like to comment.
Well, there was a key question that came across around determining the usefulness of the unbound fraction of the viral IC50 assays. And I think that's an insightful and critical question. Most of these in vitro assays are conducted in serum-free media. And many of these molecules, especially the antiretrovirals, are highly protein bound. Perhaps they have 95% of their drug is bound to protein, and therefore only 5% is available for activity. So if you were to simply look at the total drug concentration running around in the blood, and compare that to the IC50, you would find that potentially very misleading, right? Because the assays have been conducted in serum-free media. So, you know, that sort of basic understanding of how the drug operates in the body versus how the drug operates in the test tube is something that clinical pharmacology is particularly good at. And I would say that it's a blind spot, or potentially an area of errors for other people that are just really not thinking deeply through these questions. So thank you for that question, and I thought it was worth raising here so that this audience is aware of that potential gotcha.
Oh, thanks for that, Mike. Sorry, I lost our connection momentarily. So one of your unanswered questions, similar to one that you previously addressed, is about working on combination of drugs for COVID-19. I don't know if you have further comments on that.
Yeah, so it is unreasonable to assume that one of these repurposed drugs is going to be a magic bullet, right? I alluded to this at the beginning. These are repurposed drugs. They were never designed to work on SARS-CoV-2 or coronavirus ever, as far as we know. We simply don't have any treatments for coronavirus, period. So the idea that any of these ones are going to be somehow a magic panacea, I think, is stretching hope, to put it bluntly. So it's likely that this repurposing efforts will generate monotherapy treatments that have, I would say, lackluster performance. They appear to do something, but they're not in and of themselves particularly efficacious. And so I guess our thinking is, you know, going back to the battle days of our initial HIV pandemic. In those cases, we had, I would say, lackluster proteases, and we had kind of a handful of drugs that by themselves were not particularly efficacious. So we designed a cocktail, right? We went to war with the army that we had. We put together a cocktail of drugs that synergistically combined to be effective against HIV. And my thinking is that that's highly likely where we're going to end up here in this drug repurposing efforts. Over last weekend, there was a publication from a group in Hong Kong where they studied the combination of Kaletra, which is lopinavir, ritonavir. Yuan worked through that particular case, and ribavirin and interferon, so a triple or quadruple combination. And they showed that was more effective in COVID-19 patients compared to lopinavir, ritonavir alone, right? So we're beginning to see that early evidence of combination therapies being effective. We're working through viral modeling to understand where drugging different portions of the virus life cycle could be particularly efficacious. And I think those kind of silico experiments can help guide the types of clinical trials that we might like to conduct. So I really think that's probably where we're heading with this particular disease. Thanks, and great question.
Yeah, thanks for that, Mike. There were some additional questions around the determination of IC50 and the need to correct for protein binding and all of that. The questions were answered, but I was just hoping maybe other panelists or Mike could make additional comments on that. For instance, someone asked the question that could you please comment on the usefulness of determining unbound fraction in viral IC50 assays? Can we correct for that?
So maybe you could take that, because I know that we've also had some questions around exactly which cell line is the relevant choice as well. So would you mind speaking to that?
Yeah, absolutely. So we had these questions from several participants, and these are very relevant and important comments. So of course, we are taking into account unbound fractions. More data is emerging from multiple cell lines. and in vitro environments that are more relevant to the disease we are dealing with, like some cells lack an important receptor that we know plays a very important role in cell entry, and hence the need for taking into account the in vitro EC50s coming from these studies. So we are already in the process of updating the database to include multiple cell lines, like the CACO2, like Kali U3, and so on. And of notice is that results can be widely different between multiple cell lines. We even noticed some publication trying to replicate work in the same cell line, and the results were quite different. So we are looking into ways to take all of this information into account and have it summarized in a way to help with the prioritization.
Thank you, Samuel, for that. There were some questions, or at least one question about traditional medicine for COVID-19. This is of great interest in Africa, particularly in view of the information coming out of Madagascar. So I wonder if you have additional comments on that. Anyone on the panelists? Any possibility or?
Yeah, hi, this is Samir again. I typed an answer and I can reiterate here. Maybe you are aware that the WHO was holding a meeting about the subject.
Colin, do you have comments on that?
Yes,
Mike?
So Nee, this is indeed a blind spot for all of us. I think Mike used those words. Traditional medicines in this setting. Unfortunately, I don't have enough experience in that area. This particular audience might have more information. So I would say we would pose that question on the forum and see what we pull out from the combined knowledge across the African attendees in the meeting.
Yes, thanks for that, Colin. Just additional comments on that. I think part of the difficulty is the lack of knowledge about the composition of some of these traditional medications. So because we don't really know the active ingredients, and there are so many things that you need in modeling that just cannot be characterized when you have a mixture of compounds and you're not really certain which one is doing what. So that's part of the difficulty. So it will be really difficult to use modeling in that scenario. But it would be nice to see what other people have to say when we put this on the forum. And we have a very nice comment here, following up on the comment on the IC 50, correcting for protein binding from Rohan. Rohan mentioned the need to validate what has been reported in drug labels because they might be misleading sometimes. So it may be necessary for investigators to find out what the protein binding of the drug they're working on before they put it in modeling, in any models. Thanks for that, Rohan. So right now it's 4:30 West African time. That's about the end of the webinar. We still have some additional questions coming in, like we informed you at the beginning. This will be moved to the forum section of covidpharmacology.com website and will be answered. And if you have additional questions, we'll be happy to have you post them in that same forum. So once again, we say thank you to all of you who joined us today. And we are very appreciative of the effort of our panelists today who volunteered their time and contributed their expertise. I don't know if you have any final comments from any of the panelists, please, Colin, Samuel, Johan, and Mike.
No, thanks everyone. Keep the discussion going in the forums, and if you have ideas and willing to collaborate, please we need everyone's contribution to win over this disease.
So nothing else from me, Nee. Thanks a lot, everybody.
Mike?
No, the only thing I would comment is I showed at the beginning in the model-informed drug repurposing activities, I left 3 pieces unanswered, and perhaps we can commit to coming back to this group in 6 months' time or thereabouts, and we're able to fill in some of those blank spots as we learn more about this novel pathogen.
Yeah, thanks, Mike. Johan?
Thank you very much for your attention, and I do feel with our knowledge growing, with more data being connected, we're able to conquer this together.
Thank you, Johan. So like we mentioned at the beginning, if you go to Pharmacometrics Africa website, that is pmxafrica.org, you would find posts, and on that post you will have a section for this particular webinar. So all the resources that you need to do any follow-up analysis will be available on that website, as well as the recording of the webinar and the slides. So we thank you once again for joining us and wish you the very best as we conquer COVID-19. Thank you.